Well, hello, my name is Aaron Wood, and I am glad to be here. So I'll just go ahead and get right into it. So my talk today is going to be on two topics. It's going to be the development of our inexpensive fluorescence-enhancing grating-coupled surface plasmon resonance sensor platform. And the second part of the talk is going to be the testing of this aptamer lights-on fluorescence-based assay on that system, mentioned in number 1. So first of all, I'd like to start off with a brief introduction to surface plasmons in case anyone isn't quite sure what they are. So surface plasmons are free electron oscillations at a metal and dielectric interface. Those electron oscillations can be excited through the use of external EM radiation, usually through incident photons. Now, under typical circumstances, the momentum of that incident photon is not high enough to match the momentum needed for a surface plasmon resonance. So you don't get the surface plasmon resonance under normal conditions. So if we look in literature, there are two general methods of generating SPR. There's first your plasma or prism-based configurations, which you increase the momentum of this photon that travels through the prism, and it forms SPR at this dielectric metal interface. Now, unfortunately, this process typically requires bulky and expensive optical setups. The other method that they can use is grating configuration. So by using the diffraction of a photon off of this grating pattern, you can gain enough momentum to excite surface plasmon resonance. But again, there is a downside to this. The formation of these nanoscale gratings often requires E-beam lithography or interference lithography, which can be very expensive. So in order to advance this technology, as well as enhance fluorescence, we need to be able to answer this question. How do we enhance fluorescence signals using inexpensively made grating platforms for simple microscope imaging? So the way we do it is by replicating optical media. So optical media such as CDs, DVDs, Blu-rays, and HD-DVDs. These optical media, or these discs, are readily available. You can find them in almost every grocery store. And they have these regular pits and grooves. And as this technology has advanced, the spacing between these grooves has decreased. So to give you an example of this, HD-DVDs have a 400 nanometer pitch, while DVDs have a 740 nanometer pitch. So you can include a lot more data on HD-DVDs. So using these gratings, we can inexpensively produce these nanoscale gratings by replicating HD-DVD gratings in polymers. So the way we've done that is through nanoimprint lithography, which can be divided into 3 general steps. The first step being the fabrication of your PDMS stamp. So you can split your HD-DVD in half, expose your polycarbonate gratings, and pour a PDMS polymer on top of those gratings and form a negative image of them. Then, after it's cured, you can spin coat this rapidly curing ink polymer, or polymethylsilsesquioxane, or PMSSQ. And after you spin coat it on that surface, you can stamp it or put it in contact with a silicon wafer or a glass slide. And when you peel off this PDMS, you leave behind a perfect positive image of that HD-DVD in PMSSQ. Now, something interesting that happens during this peeling process is the formation of these nanoscale cracks due to stress concentrations. Now, if we take a closer look at these cracks, we notice that the nanogaps can be in width between 20 and 200 nanometers. And they're small enough that they can actually concentrate the surrounding electric field, and by concentrating that field, you can expose fluorophores in this gap and create a higher enhancement factor. So one way that we can measure the SPR, or how much light we can basically get to absorb into your grating, is by taking reflectivity measurements. We typically do this with an ellipsometer using p-polarized light. We scan spectroscopically across several wavelengths and several angles, and the resulting scan results that you get, you'll see these peaks, these sharp peaks that are indicative of SPR coupling. So at each of these wavelengths and these angles, you generate SPR. And this can also be described theoretically using this equation, which is a little bit rough there, but this left half is the momentum required for generating SPR, and this right side is the momentum of your incident photon plus the diffracted momentum. And if you plot these peak locations versus incident angle, you can develop an SPR dispersion graph, which will describe where light can couple to your grating at many different angles of incidence. So one area that I'd like to emphasize is this area for low angle excitation, so excitation at less than 10 degrees. And I'll bring it up slide after next. So another way to concentrate those electric fields, you can incorporate nanoconical structures during metal deposition. So the way that we do that is by tuning your metal deposition, such as sample temperature, your surface energy, the metal type that you're depositing, through thermal annealing, and lastly, glancing angle deposition. So with glancing angle deposition, you deposit your metal layer at an angle relative to your grating, and what this forms are extremely large nanoconical structures as well as your grating. And these nanoconical structures behave as plasmonic antennas, and they can further concentrate this electric field and further enhance fluorescence. And this can be seen in the enhancement factors of different material type or different metal types that are deposited on these gratings. So gold gratings have been shown to have a enhancement factor up to 56 times, while silver gratings can have 153 times enhancement for fluorescence, and that's due to the lower plasmonic losses of silver films. And then by incorporating these other nanostructures, you can get enhancement factors up to 173 times that of fluorophores that could be found on flat glass substrates. So this is an advantage because since you have such high enhancement factors, and if you tune your fluorophores towards low angle excitation for your SPR, you can begin to use microscopes and microplate readers to generate or excite SPR as well as image your grating platform. So this is again advantageous because imaging provides so much more information about a surface than simple spectroscopy methods. It also does not require a high-powered laser source. It doesn't require complicated optics, and they're pretty fast and easy to use. So to test this fluorescence enhancement platform for a sensor application, we developed this lights-on fluorescence assay, where we customly designed a 42 base pair single-stranded DNA chain with a thiol group located on the 3' and a Cy5 dye located on the 5'. We immobilized this DNA onto our gold grating, and under solution conditions, this DNA will form a hairpin, and due to the proximity of that fluorophore with the gold grating, the fluorescence will be quenched, or you won't see any fluorescent signal. After exposing it to a complementary sequence, or a perfect complementary sequence, the DNA will hybridize, and the fluorophore will extend up past this proximity quenching region and into the plasmonic region, where then that fluorescence will be enhanced by that electric field. So to give you some of the results from that assay, on the left we have a bright field image using a 60x water immersion lens on a gold grating. And we have enough resolution that we can begin to see the grating ridges as well as 2 nanogaps that are cutting through that gold grating. Now if we expose this grating to a mismatch sequence, the fluorescence is very low, as we would expect. But if you expose it to a complementary sequence, you can begin to see how high the fluorescence intensity is located all over this grating. So you can actually see the grating ridges and their enhancement, as well as the enhancement by these large nanoconical structures and the nanogaps that are located inside of this grating. When we exposed a flat gold surface to this, we obtained much lower intensity fluorescence. And the way that we can quantify that is by measuring the enhancement factor of this grating versus that flat gold. And what we found was that the fluorescence intensity of the grating is 13 times higher than flat gold, while the nanocones have an average enhancement of 37.5, and the nanogaps have the highest at 47.5 times enhancement versus the flat gold. So since we've had such promising results on this platform, we really want to apply it to a real sensor, as a real sensor platform, and detect things that people actually really want to detect. So we're applying it into 3 general categories. The first category being low resource setting sensors. So incorporating this plasmonic fluorescence enhancement platform with a smartphone imaging device, you can bring this device to a low resource setting and detect, say, Mycobacterium tuberculosis, or using it for water contamination testing. The second area that we want to expand into is the point of care sensor platform. So environments that have a healthcare environmental setting, and they'll have access to slightly more complicated optics. So it will enhance their detection mechanism so you can rapidly and non-invasively quantify multiple analytes. One project that we're working on at the moment is salivary hydrocortisone detection. The reason why this is important is because hydrocortisone is a biomarker for stress in patients, so you can actually quantify stress levels. The third area that we're going to be expanding into is ultra-high sensitivity lab-based sensors. So enhancing the sensitivity of lab-based sensors to the point that you can actually detect single molecules or single dye molecules with a simple epifluorescence microscope. And this is important in one area is imaging ribosome and protein conformation. So really fundamental science in how these proteins interact with their environment. So as I conclude, I'd just like to acknowledge the many contributors to this project. It would not have been possible without all of their help. First of all, my co-advisors, Dr. Shubhra Gangopadhyay and Dr. Sheila Grant, our many extraordinary lab members, our postdocs and undergraduates, as well as our international and national and corporate collaborators, and as well as our funding sources. So with that, are there any questions?